Of chapter 10, we're going to continue on with the idea of our dogma for molecular genetics of going from a protein or ending in a protein by going from a DNA to a RNA to a protein. So we left off with our RNA processing, and we're going to see how we now take the mRNA and turn it into a protein through translation. All right, so next we're going to look now at translation. So translation is this next part where we're going from an RNA to a protein. And so in translation, our ribosome translates the RNA code into a protein. So our last part of our central dogma of molecular genetics. So translation, ribosome translates RNA code into our protein. So for us to understand this, we have to understand, well, what is the RNA code? So our codex is how we're able to take the RNA code or the genetic code and turn it into something that we can read. And what you can maybe see here is, if you look up closely, that each one of our codes has 3 what we call base pairs to represent our code. And these codes here are our amino acids. So what happens is we have something we call a codon. So a codon is 3 base pairs that represent 1 amino acid. Or instead of base pairs, I'm going to say bases. And they represent a specific amino acid. Now, you're probably wondering, okay, I remember amino acids. What are amino acids again? Remember, amino acids are what we link together to create a protein. And so what we have is we have this code of 3 amino acids. So if I take one of these here, let's take this code right here, UUU. So anytime in our transcript, our mRNA, we see UUU, that actually codes for phenylalanine. So UUU codes for the amino acid phenylalanine. And so when we read our mRNA transcript, our mRNA here, we're going to read it 3 bases at a time. And as we read it, we're going to know what proteins or what amino acids link together to create a certain protein. So that's our codon, 3 bases that represent a specific amino acid. And this, our codex, gives us all the answers. So if we see a specific a codon, so 3 bases, we know which amino acid they actually represent. And we're going to see now here through translation that this is how ribosomes actually read the mRNA, by reading these 3 different codons to create our protein. All right, so let's get into that. And now let's do our ribosomes. So remember before, when we draw our ribosomes, we drew our snowman, right? And in the snowman, he had his scarf. Okay, well, that scarf is that mRNA. So that mRNA is going to leave the nucleus and come find our ribosome to create our snowman and then be translated. So our ribosome is actually made up of 2 subunits. We have our large subunit and we have our small subunit. And actually ribosomes, for a lot of the times, they are separated. So here we have our small ribosomal subunit, which literally just means it's the small part, so this small part of our ribosome. And what happens is it doesn't have the large ribosomal subunit on it yet. It's waiting for our mRNA. So our mRNA is going to come, and this little divot here is where our 5' cap is going to attach to our small ribosomal subunit. So the second reason, besides being able, well, third reason, besides stopping it from being broken down on one end and being able to leave the cell, it also lines up our mRNA perfectly with our small ribosomal subunit. Let's do this better here. See, 1, 2, 3, 1, 2, 3, 1, 2, 3, 2, 3, and we're going to make a really, really small protein. Okay, so this is our mRNA, and we're actually going to write what our codes are. So I'm going to create a protein code here really quick. All right, so here is our mRNA. And what we said before is that we're going to read off our codons to find out what amino acids go on here. So once our mRNA combines with our small ribosomal subunit, then our large ribosomal subunit is going to come in and attach as well. All right, so our large ribosomal subunit, nope, let's not do it there, it's going to be in the way. So our large ribosomal subunit will then attach. So we have our small and we have our large. Right? It doesn't really look like our snowman here, but you can kind of get the idea, the drift, and why I drew it that way. And what we find out here is that in our large ribosomal subunit, there are these indentations. There's 3 of them. We call them sites. We have our E site, our P site, and our A site. All right, E site, P site, A site. And what's going to end up happening is each one of these sites are going to be used to help read our codons. So you can see the P site is over 3 bases, so 1 codon. Our A site's over another 3, and eventually our E site will also be over 3. So probably need to make this a little bit bigger to show that. Then we're going to add another actor, and this is called our tRNA. So there's another form of RNA, and the E stands for transfer. So it's our transfer RNA. And what it's transferring is our amino acids. And our tRNA are kind of these weird looking, like upside down cactuses. And on the bottom of it, it has something we call an anticodon. So our codon is 3 bases that represent a specific amino acid. Our anticodon are going to be the opposite of those 3 bases. So if our codon is AUG, our anticodon would then be UAC. And because it's going to match up with this codon, so it's the opposite of that codon, this is called our anticodon. All right, so our anticodon, UAC, will be on whichever our codon codes for. So what I'm saying by that is, okay, so UAC, if we go back to our codex, so AUG is our first codon, and it represents methicene. We're just going to do MET. And you'll notice here that it's green. Every single protein starts with methicene. And so every single codon is AUG, the very first codon for every single mRNA. And so here our amino acid, we're going to draw this one as a block, MET, will always be attached to a tRNA with an anticodon that matches the codon. And what ends up happening is now this transfer RNA is going to transfer. Oh, that doesn't work. It's going to transfer our amino acid MET over to our ribosome, and it's going to line it up perfectly. So UAC, boom, 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 and it's going to put it inside this P site. So it's going to bring that amino acid here. So this is how our ribosome starts to translate this code, by bringing these tRNAs. Now our next tRNA, so GUC. So let's find GUC on here. Let's see, it is right here. GUC stands for valine. All right, so the anticodon for GUC is going to be CAG, and it will have, because that's got that anticodon, it's going to have the amino acid valine, so VAL. And this tRNA now is going to come over into our A site. Sorry, not the best looking tRNA here. Let me fix that up. that made it much better. And then we have our amino acid there. So once we have 2 tRNAs, each with amino acid, then our ribosome is going to unhook the amino acid that's on top of this first tRNA and bind it, create what we call a peptide bond, something we talked about, and combine it to this amino acid, and we start to make our protein. Once that happens, just kind of like an old typewriter, or like I like to say, the Beyonce song, everything is going to move to the left. All right? So our whole structure here is going to move to the left. All right, so everything is going to move this way, which is going to bring this UAC tRNA now over into our E site. Why do we call it the E site? Because E represents exit. So eventually, after it moves here, it's going to exit. Then our second tRNA is going to move over to the P site. P stands for protein, because we're starting to make our protein. It has our 2 proteins here. Or our 2 amino acids making our small protein. And that will then leave this site empty for our third tRNA to come in. All right, our tRNA is CCU, is the codon CCU. So if we go here, let's see, CCU is proline, and the anticodon is going to be GGA. And we're now going to have proline as our amino acid. This is going to come into here. So GGA. It's going to come into here to the A site. And once again, now these 2 are going to be taken off of this tRNA and added onto this protein, or this amino acid. This is why this is called the A site, because it's the amino acid site. All right? There's my blue. Now we're starting to make our protein. Then everything's going to move to the left again. I'm not going to rewrite all this. All I'm going to show here now is this amino acid moving here. So GGA. it's going to have the 3 amino acids attached to it. Making our protein. This is going to be empty again. And our last codon, which was going to be UGA, is going to come in here. So we look at what does that codon code for. So UGA is actually our stop codon. And what happens, instead of another tRNA coming in, you're going to have one of the stop codons. And the machine that comes with that, so it's going to have an anticodon of ACU. It's going to come in here, ACU. It's supposed to be a C. And what this does is this says, guess what? Our RNA is done being translated. We've made our protein. Our protein has just these 3 amino acids in it. And this tells the ribosome we're all done, and then everything detaches. The large ribosomal subunit attaches, the small ribosomal unit detaches, this detaches, our protein detaches and goes into the ER, and even our mRNA detaches. But then that mRNA, as long as it still has time left on its stamp there, can go find another small ribosome and go through the whole process and make more protein. That is translation. That is how our ribosomes are able to read off the code that's found in these codons by using these anticodons on the tRNA. Those tRNAs will bring in the corresponding amino acid, and then our large ribosomal subunit will start to link those amino acids together as we read off this code. Now there's no protein out there, there's only 3 amino acids long. So these mRNAs, usually their smallest protein has at least 100 amino acids in it. So obviously we weren't going to do something like that. That would take forever and just make our board look completely messy. But you can see here now that this is how we go from a DNA by going through transcription, processing that transcribed RNA into an mRNA. The mRNA comes out, finds our ribosome. The ribosome then translates our code using the help of our codons and our anticodons and our tRNAs, bringing in our amino acids, then to create our protein. So we have now fulfilled this central dogma, going from a DNA to an RNA and a protein. Now, what happens when things go wrong, right? That's what we're going to talk about next. All right, so when we're looking at DNA replication, DNA transcription, DNA translation, the whole idea is that everything is happening like it's supposed to. Well, there are times, and most specifically, the biggest problem is when we have things going wrong in our DNA replication or synthesis. If we don't make an exact copy of our DNA strand, then we can run to things we call mutations. And mutations, we often refer to these as the spice of life. All right, and we'll talk about the reason why that is. But mutation is any change in the DNA sequence. So if we had A, T, C, G, G, A, T, C, and then T, A, G, C, C, T, A, G. So that's our original. If we were to change, if we made a copy and we ended up changing that C into a G, now this is a C, and we have now had a mutation occur. All right, so that can happen when we're replicating our DNA. That can happen when there's outside forces that come in. So like sun is one of those things that comes in and will break up your DNA. So when you go to repair your DNA, you might have some mistakes on that. Other things that cause cancer, like carcinogens, all that kind of stuff, also can have effects on your DNA, and that's where we get our mutations. So what we often will call this the spice of life, and the real reason for that is if we think about life as a single cellular organism that started in oceans 4-point-something billion years ago. If we never had mutations, we would all still be single cellular organisms in the ocean, or maybe not even be here anymore because we wouldn't be able to adapt to our environment. So mutations, even though most of the times they are bad, they're deleterious, they're harmful, they kill the cell, every once in a while you get a mutation that's actually beneficial, and those mutations, as they accumulate over millions and millions of years, is how evolution is actually able to occur. And we'll be talking a lot about evolution in some later chapters down the road. Sorry. So that's why we call it the spice of life. So mutations are, like, if we look at them individually, they're bad because most of the time it kills the cell. But if we look at whole life as a whole, it is the spice of life. It is what makes life as diverse that we see it now, from all the different animals, plants, insects, bugs, fungi, bacteria, viruses. All that is based off of mutations, that spice of life. Just like when we have food, we add spices to it to make it taste different, to give it different flavors. That's why we call this the spice of life. All right? So even though we're going to be talking about mainly how this is bad, realize in the big picture, without mutations, we aren't here. All right, so we have two different types of mutations that occur. The first is what we call a base mutation. And this is where any one of your single bases, just like we did here, gets changed out for something else. So we have three different examples of this. We have missense, so the change of a single base to another. And you're wondering, okay, well, how does that have an effect? If we go back to our code here, right? We see that we have these codons that go for a specific amino acid. So if we had a codon that was UUU, so in other words, in the DNA code, UUU would be AAA. So that means this would be TTT. Well, what would happen if we changed it up? So instead of it eventually becoming UUU, it became UCU. So that this got changed over to a G, change that to a C. Now this codon, instead of being UUU, it's going to be UCU. And what happens is that now changes our protein from having a phenylalanine here, it's going to have a serine instead. And that can have then an effect on how that protein works. Now, if you ever heard of sickle cell anemia is based off of a missense change. What happens in sickle cell anemia, our normal blood cells are circular like this. And because they're circular like this, inside they have something called globin, hemoglobin. And that hemoglobin is actually what carries our oxygen to our cells. And it also carries our CO2 out of our cells. Because that's all that blood does. The blood's, one of its main purposes is to take oxygen from our lungs and just distribute it all over our body to our cells. And as it's taking the oxygen, it's picking up the CO2, and then when it comes back to our lungs, it releases the CO2, picks up more oxygen, and goes back through our body and does that. That's what our blood cells do, right? So there's a protein inside our blood cells called hemoglobin that does that. So when we have a missense mutation, so a base substitution, in our hemoglobin, it changes the shape of our blood cells from circular to sickle cell type, or like a half moon. So a sickle, so think of Death, and he carries a sickle. Sickle is used to cut down, to harvest grain and stuff like that. That's why it's called sickle cell. Now, sickle cell anemia, what that does is it makes it harder to carry oxygen and CO2. So people have sickle cell anemia if they live in high elevation places where the oxygen levels aren't as saturated. They have a harder time breathing, and they can actually die. And so people who have sickle cell anemia, you will often usually move down by the coast or into low elevated areas where there's higher oxygen concentration. But the other problem with sickle cell anemia is it causes plaques inside your blood. And the reason for that is these sickle cells actually can connect to each other like this, and that's what causes those plaques. So they end up building up and doing big plaques and clog up your arteries, and then you can die from that as well. And so that's why sickle cell anemia is very dangerous, and people who have it usually will get a bone marrow transplant to reset their stem cells to be able to create regular blood cells once again. So that's a missense where you're just changing one base pair, but it has a downstream effect to where now it changes a protein structure because it changed one amino acid, and just like sickle cell anemia, it can have disastrous effects to that individual. Now, silent mutation is when you have that change. So let's say this is supposed to be AAA. And instead of changing that to a C, you end up changing this one to a C, or sorry, a G. So that's the change here. So this is now a G, and this is now a C. So now the codon here is going to be UUC instead of UUU. Well, if we look at it, UUC also codes for phenylalanine. So therefore, it's a silent mutation, because even though it's a mutation because we changed our DNA sequence, it's silent because it didn't affect our phenotype or our protein. We still coded for the same amino acid. So that's a silent mutation. Anytime there's a mutation, but it doesn't actually affect the protein or the amino acid sequence. Then we can have a nonsense mutation. All right, so let's say this got changed to UCC. So we changed those two. Now instead of this coding for UUU, it now codes for UGG. And if we see, oh, not UGG, I meant the wrong one. So these two get changed. So now instead of being UUU, it's UGA. And we see UGA now is a stop codon. So that stop codon, what that does is that stops the protein early. So maybe that was only the 100th codon, and there was supposed to be 300 codons because it was supposed to be 100 amino acids long. Now it's going to truncate and make that protein only 30 amino acids long, and now you have a small protein. So these nonsense ones are pretty big when they happen because they can change a huge effect, have a huge effect on the size of the protein that is being created. All right, so that's when we have a base substitution. Now we can do it where we have a base addition or subtraction. So in other words, if we were to add in another base here, so we added in, so let's go back to these being regular. but you ended up adding in another base here, so let's make it a C. Well, this is still U, then it was supposed to be C, U, but instead it's going to be GCU, and you can see that's going to make a difference. So instead of CCU, it's going to be, where's GCU. So we've already messed up one, but then it's going to mess up every single codon downstream. And so anytime we add a base or subtract a base, it's going to affect every single codon. It's going to make it a completely different protein from that mutation on. So if this is near the beginning, then it's going to have a huge problem. Or even if it's in the middle or the end, it's going to change up how that protein is. And it's at that point also probably going to introduce stop proteins earlier, so it really has a strong mutation or effect on the protein down the line. That's why most of these mutations are damaging or lethal to the cell, but every once in a while you get that good mutation, the environment acts on it, and then you can have these different type of evolutionary things that come about from our mutations in that spice of life. All right, that is the end of chapter 10. For our next set of chapters, we're going to be talking about biotechnology. So this is going to be chapters 11 and 12. We won't go as much. There'll be some things in the book that we're talking about, but really we're going to talk about some cool, fun things. So one of the things we're going to be talking about are like cloning. How did cloning get started? What's going on with cloning these days? We're going to talk about the coronavirus. We're going to look at how it works as a virus. We're going to also talk about HIV and how there's people out there that are resistant to HIV. And then we're also going to be talking probably about stem cells and how stem cells work and how they could help with the medical profession in the future. And then we also might talk about the possibility of how we could end up accidentally creating zombies as well.